So the issue of the recorder is not my fault. I don't know what happens. It only seems to happen to me, but the discs will go unformatted. And then I don't know how to fix it. Then I have to get to Charlotte, and then Charlotte reformats it. And then it works. So I don't know, maybe it's karma. Maybe I've done something to the environment of the world that then ultimately subsequently impacts you guys. But at least the recordings are posted from last year to be able to access the Tuesday's lecture. So, continuation on of Tuesday's lecture, we will start where we finished off, which was we talked about some of the respiratory mechanical principles, so relationships between airway resistance and lung volume, flow volume loops, spirograms, pressure volume relationships. I had you take breaths into you were full, try to breathe at total lung capacity, breaths out to you're empty, try to breathe the residual volume. And you got to experience the perceptual consequences of breathing against high resistance and high elastance. Giving you an appreciation for why it's very important to try to breathe along the linear portion of the pressure volume curve. So, again, those are some basic kind of static and dynamic mechanical aspects of breathing. In the context of exercise, there are some challenges that the respiratory system faces. We'll discuss those, move into some aspects related to the control of breathing during exercise, the physiological determinants of ventilatory demand during exercise, and then ultimately in some of the subsequent lectures looking at some experimental evidence of what's really contributing to the control of ventilation, which is a very complex and actually pretty challenging thing to study because the respiratory system and all of the signaling mechanisms are actually very redundant, which is to say that if kind of one signaling pathway goes away, other ones are able to seemingly compensate, and ventilation seems to, for the most part, be really well matched to metabolism, even when you seem to eliminate critical sensors and receptors for the control of ventilation. So I'll provide you some experimental evidence of all of that. So what is the challenge faced by the respiratory system and the lungs and the respiratory muscles and airways? And so we know that during exercise, there are exercise intensity dependent increases in the rate of oxygen consumption and carbon dioxide production. And so really, the lungs are the conduit in which fresh air, so higher levels of oxygen are basically transported to exchange at the lungs, and carbon dioxide that's produced at the tissues are exchanged to be expired. So we have basically delivery of oxygen, which now we have a pretty strong appreciation for the impact of oxygenation at the cell on the rate of development of muscle fatigue and exercise performance. And at the same time, we know that carbon dioxide has the potential to liberate hydrogen ion and therefore make the body acidotic. And so ventilation is a means of regulating acid-base balance in very rapid kind of swings, right? Acid-base balance disturbances would occur, for example, right now if you held your breath, CO2 levels would rise, hydrogen ion would increase, that would stimulate chemoreceptors, and part of the reason you can't hold your breath until you pass out. At the same time, you could voluntarily hyperventilate now and blow off carbon dioxide and shift various mass balance relationships, and you would get rid of hydrogen ion, and so you would become alkalotic. So you'd have a pretty severe respiratory alkalosis. And again, the respiratory system is a means of causing pretty dramatic swings in hydrogen ion relationships due to changes in what we do. Either we retain carbon dioxide or we blow it up. And so the first challenge ultimately is that exercise-induced increases in the rate of oxygen consumption and carbon dioxide production cause the blood that returns to the lungs from the exercising muscles, which again we call the mixed venous blood, to get progressively more hypoxic and hypercapnic. So you can imagine then that you've got the arterial blood is being sent to the muscles. They are stripping increasingly more oxygen off, so the amount of oxygen that's returning back to the lungs has a progressively decreased partial pressure of oxygen. At the same time, carbon dioxide is being loaded from the muscles into the blood to be just sent back to the lungs to be blown up. And so we basically have this challenge where we need to, in an attempt to try to protect arterial oxygenation and gas exchange and prevent hypercapnia, the lungs really need to facilitate increases in ventilation to try to bring more fresh gas in and eliminate carbon dioxide from the body. And so ultimately, this disturbance or these changes in the rate of oxygen consumption and carbon dioxide production have the potential to disturb acid-base balance and also compromise the delivery of oxygen to the exercising tissues, which we know or now have an appreciation can lead to fatigue through various biochemical and bioenergetic pathways. Second, the respiratory system has to kind of meet these increased metabolic requirements that can rise, depending on your level of fitness, 20 to 30 times above rest, while maintaining or minimizing the mechanical and the oxygen cost of breathing. So again, this is where it becomes very important in terms of how we breathe and where we breathe along that pressure volume curve, right? Because remember, the change in pressure relative to the change in volume dictates the work of breathing, and a higher work of breathing will require a greater oxygen cost. A greater oxygen cost of breathing would require more blood flow. More blood flow would require a greater percentage of an available cardiac output. If more cardiac output is going to your respiratory muscles and not your quadriceps will have less oxygen for a given demand, and therefore you would be able to go through all the series of events that would result in a faster degradation of phosphoprotein, creatine, greater accumulation of inorganic phosphate, hydrogen ion, greater reliance on, say, glycolytic flux to support a given amount of ATP development, greater rate of development of fatigue, right? So again, these are kind of those series of events that hopefully you are able to start seeing the links whenever we talk about something that can compromise oxygen delivery. For example, an elevated work and oxygen cost of breathing. So ultimately, this minimization of the mechanical work of breathing requires that the ventilatory response to exercise increases in direct proportion to muscle metabolic demands, particularly carbon dioxide production. We actually ventilate in many which ways to control carbon dioxide levels. We don't really ventilate, what people don't believe is or they're not really familiar with, is that we don't really ventilate to control oxygen levels. We more in large, we ventilate in a way of trying to control CO2 more than anything else. And a great example of that, one that usually anchors it for people, is that if you, say, held your breath right now, just held your breath. Most people, between subject variability, could do it 15 seconds to 90 seconds, okay? Now, this is here, you're at sea level, your blood is completely saturated with oxygen, you got 97% oxyhemoglobin saturation. So let's say, for example, that prior to another voluntary hyperventilation procedure, a voluntary breath hold procedure, you voluntarily hyperventilated for, say, 3 or 4 minutes. This is what free divers do, right? So the voluntary hyperventilation has what effect on oxygen levels in your blood and your oxygen saturation? Really have all that much? I mean, your blood is already fully saturated. You're not really driving much in the way of an increased partial pressure of oxygen into the blood to increase the dissolved component. And so hyperventilation prior to, say, a voluntary breath hold procedure really doesn't do much to your arterial PO2 and your blood saturation, your oxygen content. What it does do is that it increases ventilation above and beyond metabolic production of CO2, and it blows off a ton of CO2. So you become very hypocapnic and alkalotic. So now if you try to do a voluntary breath hold after 3 or 5 minutes of prior hyperventilation, you can hold your breath 3, 4, 5, 6 minutes. So the point is that it's not the oxygen, there's no change in oxygen, but nevertheless you can hold your breath for 2, 3, 4-fold more. It's actually the blowing off and the change of CO2 that allows you to actually hold your breath, because oxygen levels are not changing with prior hyperventilation, it's CO2. So it's kind of just a very simple example that your ventilation is largely regulated to try to control CO2 levels because it has the most potential impact to disturb acid-base balance. Yeah?
No, I was wondering if you were trying to, like, on the dissociation curve, or more like on the, not on the same one, but I guess.
Yeah, but again, I mean, you wouldn't really be moving down or up that curve because at that point it's pretty linear. And the second component is that we really have to be precise about the way that we control our operating lung volume. So from the previous lecture, it's those end expiratory and end inspiratory lung volumes, and the difference between the two that dictates our tidal volume. Right? So it's the end inspiratory and end expiratory lung volume that really dictates where we're breathing in relation to total lung capacity and residual volume. And I'll show you later on some evidence of how most people choose to breathe or they do breathe in the context of exercise. And third, another challenge is that the work produced by both the locomotor and respiratory muscles increase several fold during exercise. So as you can imagine, blood flow and oxygen requirements of both muscle groups must be met. And any circumstance where there's an excessive increase in the work and an oxygen cost of breathing has the potential to, for lack of a better explanation, steal blood away through sympathetic mediated vasoconstriction from the exercising muscles, so like the quadriceps, the glutes, the biceps, whatever it is, leading to peripheral muscle fatigue. And so we'll discuss this later, but it's interesting. The respiratory muscles, particularly the diaphragm, don't have a ton of innervation in terms of alpha-1 and alpha-2 adrenergic receptors. So what happens is that when there is an activation of the sympathetic nervous system, the diaphragm, for example, is relatively is poorly sensitive to muscle sympathetic or increase in sympathetic nervous system activation in terms of vasoconstriction of its blood flow. So what happens is that if the respiratory muscles become burdened and send basically a signal to activate the sympathetic nervous system, it has a preferentially greater vasoconstricting effect on the quadriceps or the exercising muscles than it does the diaphragm, because there's not many alpha-1 and alpha-2 adrenergic receptors in the diaphragm. So again, vasoconstricting influence, but if you don't have receptors, you don't really have an impact. It's just like you can go onto a putting green that doesn't have a hole, and you can putt the ball around, it's never going to go into a hole. You're just hitting a ball in a green, you're not hitting it in there, right? So if it doesn't have receptors, the ball has nowhere to go. This influence has nothing to kind of affect. So, what are some of the determinants of arterial blood oxygenation? You've seen this before. So, diffusive oxygen flux across the lung is really a reflection of the diffusive conductance of the lung for oxygen multiplied by the pressure gradient between the lung, so the alveolar partial pressure of oxygen, and the pulmonary capillary partial pressure of oxygen. So basically we have the permittance of oxygen flux across the lung and the driving pressure. So we can, to get an increase in diffusive oxygen flux at a given level of conductance, we can increase the driving pressure. We could increase the conductance at a given driving pressure by, say, increasing the surface area for gas exchange or any combination thereof. Impairments in oxygen flux obviously could be due to opposing effects on these parameters. So these determinants, so the diffusive conductance of the lung for oxygen is really dictated by the surface area for gas exchange. So again, we talked about this, that in a healthy individual we have a massive surface area for gas exchange, in large part because of the honeycombing effect of all of those very, very small alveolar units. And the diffusion distance that exists between basically these alveolar units and the pulmonary capillaries. And so, fortunately, we have a massive surface area and a very, very small or short diffusion distance. So the net effect is that we have a very high diffusive conductance for oxygen across the lung. So, anyway, oxygen is pretty permissive to move from the lung into the blood. Right? So one of the major determinants then of exercise-induced facilitation of oxygen exchange is really changes in the driving pressure, or the gradient, for oxygen flux from the lungs to the blood. And so the pressure gradient for diffusion of oxygen is dependent on the alveolar PO2, which is basically the high energy component of oxygen flux. So this is going to be the partial pressure of oxygen in the lung tissue, which in turn is determined by alveolar ventilation. So alveolar ventilation, as you see, is not necessarily the same as minute ventilation. Alveolar ventilation is the amount of ventilation that occurs actually at the level of the lung. It's an index of basically how much fresh gas is delivered to the lung. And alveolar ventilation is dependent on physiologic and anatomic dead space as well as your breathing pattern. Okay? Minute ventilation is a function of basically the depth of your each breath and the frequency that those breaths are taken. So basically, minute ventilation is the amount of air you move into and out of your mouth or into your total respiratory system. Alveolar ventilation is how much ventilation occurs at the actual lung. And depending on breathing pattern, the matching of the two is better or worse. If you breathe deep and slow, the matching of the two is better. If you breathe fast and shallow, the matching of them is very impaired. So again, the nature of your breathing pattern and the one that you adopt during exercise does have an influence on gas exchange. So the statement here is that if alveolar ventilation is inadequate, so let's say for example you have an individual with disease, where it's not that they don't want to breathe, but they physically can't breathe due to, say, airway obstruction or restriction, as an example, is that if alveolar ventilation is inadequate, particularly for metabolic demands, the partial pressure of oxygen in the lung will be inadequate to ensure that the rate of diffusive oxygen flux from the alveoli into the blood is able to fully saturate the red blood cells with oxygen during the brief time that these red blood cells actually spend in the pulmonary capillaries. Okay, so we need a high driving pressure to try to facilitate rapid exchange of the oxygen from the lungs into the blood to bind to the red blood cells and diffuse into the plasma in order to be delivered back to the exercising tissues. Okay? So this becomes increasingly more important and a challenge during heavy exercise, since the amount of time for pulmonary oxygen gas exchange progressively decreases due to reductions in both mixed venous PO2 as well as the time the red blood cell spends in the pulmonary capillaries. So with increasing cardiac output, you get more rapid so shorter transit times of the red blood cell through the pulmonary capillaries, so they don't spend as much time at that lung-blood interface. So really, to facilitate the gas exchange with that more rapidly moving blood flow, we need to widen that pressure gradient. So the reduction in mixed venous PO2 is one way to certainly facilitate that pressure gradient, right? Because we'll have a reduction in pulmonary capillary PO2 as our body begins to consume oxygen and the mixed venous blood gets progressively more hypoxic. But if we also increase ventilation, alveolar ventilation in particular, adequately, is that we can increase to some extent the alveolar partial pressure of oxygen, which further will widen the gradient and facilitate gas exchange to try to offset that shortening of the red blood cell transit time. And for the most part, these adaptations are very effective. And you don't have much in the way of dramatic disturbances in oxygen exchange. So the determinants of pulmonary diffusive oxygen flux really conform to Fick's law of diffusion, which I'm sure you're all familiar with. And so according to Fick's law of diffusion, this rate of diffusive oxygen flux from the lungs to the blood is proportional to the surface area, the diffusion coefficient of the gas, which basically is a function of the solubility and size of the gas molecule. So carbon dioxide, for example, is a much more soluble gas than oxygen, and so carbon dioxide moves much more freely from the blood to the lung than oxygen does from the lung to the blood. Again, it's just a function of the solubility of the gases at a given partial pressure. Of course, we have the driving pressure, so the alveolar to capillary gradient for oxygen, as well as the diffusion distance. So you can appreciate that really, for the most part, three of these factors are relatively fixed or established. They're probably not changing all that much in the rest exercise transition. You know, you might get you have a relatively constant surface area. You might get some changes with the recruitment of pulmonary capillaries or whatever it is, but for the most part, surface area for gas exchange is probably relatively fixed. The diffusion coefficient of a gas is going to be constant. You're breathing oxygen, that's going to be relatively constant. The diffusion distance, so unless your lung gets wet and you develop pulmonary edema due to some type of like hemorrhage due to very large forces that are placed on the lung, which often happen in race horses and things like that, because they basically generate so much blood flow and they get this capillary interface failure where the blood vessels will actually kind of explode and then the lungs will get wet, and that compromises the diffusion distance, so it increases basically the thickness for gas exchange, and so horses will develop incredibly severe hypoxemia during races, and oftentimes they'll bleed out of their nose with basically this blood coming from their lungs, because they have such a massive capillary stress failure, because they have so much muscle relative to relatively small lungs. So it's an interesting animal in terms of its size characteristics in the respiratory system. And a major factor is the driving pressure. So you can appreciate that from this Fick's law of diffusion, anything that increases the surface area for gas exchange modifies increases the diffusion coefficient, will widen the pressure gradient, will increase the rate of diffusive oxygen flux, and anything that will decrease the diffusion distance will increase the rate of diffusive oxygen flux. Alternatively, anything that will have an increase the diffusion distance will negatively affect the rate of diffusive oxygen flux, with the opposite effects occurring in terms of gas exchange if you if you decrease surface area, had a less diffusive gas, and you narrowed the driving pressure. Right? So, this is a bit of a, to say is a bit of a busy slide would be an understatement, but what we're doing is we're trying to bring back in now the conservation of mass model to understand bringing in the inflow and outflow model to understand the delivery of oxygen basically into the lung space, okay? So what's determining the alveolar PO2, right? So here we have, again, we got that compartment model. Remember we talked about where we had cardiac output and total vascular conductance determining mean arterial pressure, right? So mean arterial pressure was really the volume of blood that existed within the systemic circulation. What we're talking about now is we're talking about the oxygen volume within the lung. So the compartment now is the lung. And the inflow is now going to be determined as alveolar ventilation. Okay, so how much fresh gas is how much fresh oxygen is delivered into that compartment. And the outflow, pulmonary oxygen outflow, is really determined by pulmonary blood flow. So we have ventilation, which is the determinant of how much oxygen is being brought into the lung, and pulmonary blood flow as a determinant of how much oxygen is leaving the lung. Okay, so again, we come back now to our inflow and outflow model, our conservation of mass model. So, pulmonary inflow is again determined by alveolar ventilation, which is really a function of minute ventilation, so how much air are we moving into and out of our body per minute, minus the dead space ventilation. So we have a physiologic and anatomic dead space that occupies a certain amount of each breath, every tidal breath. So again, we have, for example, the non-diffusible portions of the lungs. What is it, from generation 0 all the way down to 13 or something like that? We looked at the anatomy of respiration. It's all of those non-diffusing components of the respiratory system as an example. If you didn't see the lecture recording from last time, that wouldn't make any sense. So, but you have a diffusible portion of the lung and a non-diffusible portion of the lung. The non-diffusible portion of the lung basically represents a dead space, right? It's just gas that's kind of trapped in the respiratory system. So you need, for every single breath, you need to ventilate that amount of trapped gas that doesn't participate in gas exchange. So there's a fixed dead space there. So not every breath is perfectly efficient because we don't have a system where every centimeter of a respiratory system participates in gas exchange, okay? So, the diffusive conductance of the lung for oxygen, again, is a function of the surface area and the diffusion distance, which we said were large and very short, respectively. And the outflow, again, is determined by pulmonary blood flow, which, again, is going to be determined in large part by cardiac output and local vascular resistances in the pulmonary capillaries. And so, again, pulmonary diffusive oxygen flux is equal to the diffusive conductance of the lung divided by the driving pressure. So as it states here, alveolar ventilation represents the inflow of oxygen or fresh gas into the alveoli, while movement of oxygen from the alveoli into the blood, and the flow of the blood out of the lungs represents outflow. The balance between alveolar ventilation or inflow and pulmonary blood flow outflow will ultimately determine the alveolar PO2. So if we have a disproportionately high pulmonary blood flow relative to alveolar ventilation, our outflow is greater than our inflow. So what's going to happen to the alveolar PO2? If outflow is greater than inflow, what happens to compartment volume, pressure, or concentration? It's going to decrease. So if we decrease the alveolar PO2, what happens to the driving pressure for oxygen diffusion across the lungs of the blood? It's going to decline, right? So you can appreciate that we need to have some matching. We don't really want to have, it's better to have disproportionate increases in alveolar ventilation than it would be in, say, pulmonary blood flow. Because then that would actually increase the alveolar partial pressure of oxygen, which would facilitate oxygen exchange between the lungs and the blood. We wouldn't necessarily want to have a circumstance where pulmonary blood flow is disproportionately high for alveolar ventilation, because we'd be creating a circumstance where oxygen outflow is greater than oxygen inflow into the alveolar compartment, which would not facilitate gas exchange. So, the point here is that any constraint on alveolar ventilation has the potential to limit the ability to maintain a high enough alveolar PO2 to to facilitate gas exchange at a given level of diffusive oxygen conductance for the lung. And this has the potential to compromise arterial blood oxygenation and oxygen supply to the exercising muscles. So again, if our oxygen saturation, our partial pressure of oxygen dissolved in the blood, and ultimately the arterial oxygen content is compromised by some type of impairment in the relationship between alveolar ventilation and pulmonary blood flow, is we have a circumstance now where, remember back to the previous experiments where we manipulated at a given muscle blood flow in the animal model, at a given muscle blood flow, they changed arterial oxygen content, and there were changes in phosphocreatine degradation and inorganic phosphate in order to maintain a given level of force production. So you can appreciate then that if you change any one of these or combination of these factors, you have the potential to change the amount of oxygen that's being delivered to the cell at a given rate of muscle blood flow, and therefore you would change, presumably, intracellular PO2, which would have negative effects and consequences on compensatory mechanisms to support a given amount of exercise. So, the idea here then is that matching alveolar ventilation and pulmonary blood flow to maintain or result in an elevated alveolar PO2 is very important to facilitate oxygen exchange and maintain a sufficient rate of oxygen delivery to the peripheral tissue, which again is a function of muscle blood flow and arterial oxygen content. So it's really influencing the extent to which we're impacting on arterial oxygen content. So, why is it important to match alveolar ventilation to metabolism? So here we have a couple of other equations that you should familiarize yourself with. So during exercise, again, the respiratory system is faced with the challenge of maintaining alveolar gas exchange by protecting both alveolar PO2 and alveolar partial pressure of carbon dioxide. And this occurs through the precise matching of alveolar ventilation to muscle metabolism. Most notably, the rate of development or production of carbon dioxide by the active, primarily, and to a lesser extent inactive tissues. So here we have the alveolar partial pressure of carbon dioxide and the alveolar partial pressure of oxygen. The alveolar partial pressure of carbon dioxide can be explained by the relationship between how much carbon dioxide is produced by the body relative to our total alveolar ventilation. So the rate of ventilation at the level of the lung, multiplied by the barometric pressure minus water vapor pressure. And so really what you're seeing is that we have a relationship between the alveolar CO2 and the arterial CO2, such that if alveolar CO2 levels rise, we're going to get a rise in arterial CO2 levels because we're modifying the pressure gradient for exchange that will facilitate basically CO2 remaining into the arterial blood and not exchanging across the lung. From the blood, mixed venous blood, into the lung to be expired. If you look at alveolar PO2 is equal to the inspired partial pressure of oxygen, which is really just the percentage of oxygen in the inspired air multiplied by the barometric pressure, taking into account the water vapor pressure. Because water vapor itself imposes a pressure in a particular substance, minus the partial pressure of carbon dioxide in the arterial blood, divided by the respiratory exchange ratio, so how much carbon dioxide is being produced relative to oxygen being consumed. And ultimately, you see a relationship then between the alveolar partial pressure of oxygen and the amount of oxygen in the arterial blood in terms of saturation, the amount of oxygen dissolved in the plasma, and ultimately the oxygen carrying content, or the oxygen content of the arterial blood. So let's say, for example, that at the onset of exercise, we know that our cells will begin to produce carbon dioxide, okay, as a byproduct of metabolism. So carbon dioxide elevates. Now, if there is not a proportionate increase in alveolar ventilation, what would happen to the partial pressure of carbon dioxide in the alveoli? It's going to rise. So if you have increased concentration or partial pressure of CO2 in the alveoli, what's going to happen to the arterial CO2 levels? They're going to rise. Now, what is the impact of CO2 retention or carbon dioxide retention in the arterial blood on alveolar PO2? Well, you can see is that if you do not increase alveolar ventilation in proportion to metabolic demand, and you get a rise in alveolar and arterial CO2, this will actually have an impact on the amount of oxygen that exists in the lung, because there is ultimately a certain amount of pressure that's exerted by carbon dioxide and oxygen within a particular space, as you'll see later. And so if you increase the arterial CO2, this actually has the effect of decreasing the alveolar PO2, which has the consequence of actually decreasing or compromising arterial levels of CO2, whether or not bound to hemoglobin, dissolved in the plasma, or the total oxygen content. So what we need to do ultimately, at the very least, is that we need to have a system or a circumstance where alveolar ventilation is able to increase in proportion to the rate of metabolic production of CO2. This will maintain or restrain the rise in alveolar CO2 levels, which will maintain arterial CO2 levels at or near resting values, and this will allow the alveolar PO2 to remain relatively preserved in the rest to exercise transition, and therefore we don't have much in the way of disturbances in arterial oxygen levels. So you can appreciate then in this particular example that it's really alveolar ventilation, and through its matching to the metabolic production of carbon dioxide, that ends up influencing the PO2 in the lung. We're not ventilating directly to regulate or control oxygen levels. It's just the oxygen levels regulation is circumstantial through ventilation's control of CO2. Okay. So, looking at some of the factors of pulmonary gas exchange, we have to have appreciations for minute ventilation, anatomic, and physiologic dead space. So as I mentioned previously, minute ventilation is the volume of air moved into and out of the lungs per minute. So this is kind of like bulk flow. This is not telling you about the efficiency of that ventilation, it's just telling you about ventilation, okay? The total movement of air. But it's not necessarily telling you about how much air is being delivered to the lung to participate in gas exchange. So minute ventilation is the product of tidal volume expansion, so the volume of every breath multiplied by your respiratory rate or your breathing frequency. So then there's two forms of dead space. There's an anatomic dead space and a physiologic dead space. Anatomic, think of it in the way is that it's something that's kind of fixed. It's anatomically defined. So somebody that's very, very tall, for example, and has longer airways will have a larger anatomic dead space than somebody that's very short, right? I will have a larger anatomic dead space than a baby, because I'm big. So it's anatomically defined. So this is really the volume of air that fills the non-diffusible portions of the respiratory system. So those conducting zones that I showed you, I think in the first slide of the last lecture. So the nose, the mouth, the trachea, the bronchi. In healthy individuals, the anatomic dead space is about 30% of every tidal volume. So 30% of every tidal breath is basically gas that does not participate in gas exchange. Okay, so we have an inherently somewhat inefficient system because we have to ventilate these non-diffusible portions of the lung. This is relatively unchanged. It's pretty much fixed. Ironically, during exercise, your anatomic dead space can actually increase because the conducting airways can actually relax, the airway smooth muscle can actually relax and they can bronchodilate, and the anatomic dead space during exercise can actually increase a little bit with exercise-induced bronchodilation. Fortunately, it's not to the point where it's ineffective, like it has a negative impact because the other ventilatory adjustments are of much greater magnitude. Then we have the physiologic dead space. So the physiologic dead space is the portion of alveoli, or kind of the percentage of alveoli that are either poorly ventilated and or poorly perfused with blood. So this is where we get into ventilation-perfusion relationships. We can have a lung that has, say, great surface area for gas exchange. It looks perfect under a microscope. It has nice honeycombing, but let's say that you had some type of pulmonary vascular disease. So here you have a lung that's well ventilated but poorly perfused. That would serve to increase physiologic dead space. You can have another circumstance where you have a deteriorated lung in terms of, say, severe emphysematous destruction with these enlarged alveolar units, but you could have a perfectly normal cardiac output and pulmonary blood flow response. The net effect is that either whether or not it's it's a poor ventilation or poor perfusion, or some likely, most often in disease, some combination of the two, is that you have areas that develop high ventilation perfusion ratios. So again, areas that aren't really participating much in the way of gas exchange. Fortunately, in healthy individuals, this is a relatively negligible component, so about 250 to 500 mLs, but this will tend to increase in adults with cardiopulmonary disease. So again, if you have emphysema, pulmonary vascular disorders, if you have, say, heart failure and impairments in pulmonary blood flow, all of these factors can contribute to an elevation in physiologic dead space, which effectively needs to be compensated, as I'll show you, by increasing ventilation. So alveolar ventilation then, alveolar ventilation is the volume of air that reaches the alveoli and participates in gas exchange per minute. The alveolar ventilation is equal to the minute ventilation, which again is the product of tidal volume expansion and breathing frequency. Yeah.
On the earlier slide, in physiological dead space, is it 0.25 to 0.5 liters? On the bottom.
Sorry, yeah, sorry, 0.25 to 0.5 mLs, not 250 to 500 mLs. Yeah. So the alveolar ventilation is equal to the minute ventilation minus the dead space ventilation. So we know that minute ventilation is the product of tidal volume and breathing frequency, but the dead space ventilation, again, this is a rate, okay? So anytime you basically see like V dot A, V dot O2, V in this case apostrophe A, because instead of going in and putting in text boxes and aligning the dot over top of the V, which is far too time consuming, the apostrophe next to the V represents a rate, which means that it's some unit over time, okay? So the dead space ventilation is the volume of air that does not participate in gas exchange per minute, where the dead space ventilation is the sum of the anatomic and physiologic dead space multiplied by the breathing frequency. So you can appreciate then that if you had an elevation in physiologic dead space due to, say, emphysematous destruction of the lung or some type of impairment in pulmonary blood flow, you would have, and also a circumstance where you had to increase breathing frequency. Let's say that you had some type of obstructive or restrictive impairment in breathing mechanics and you couldn't breathe deeply, so you had to rely on breathing quickly. You would have a very elevated dead space ventilation. So what would that do to alveolar ventilation? So let's say, for example, that you were breathing at, say, 50 liters a minute of minute ventilation, but then you had a circumstance where the anatomic and physiologic dead space was elevated, and that minute ventilation was achieved not by breathing deep and slow, but by breathing rapid and shallow. What would happen to dead space ventilation under those circumstances? Dead space ventilation would be quite high. Dead space ventilation would approach the value of minute ventilation, right? So let's say, for example, you had a minute ventilation of 50 liters a minute, but a dead space ventilation of 40 liters a minute. The alveolar ventilation then would only be 10 liters a minute. So really, you're operating at what, like 20% efficiency. For all of the air that you're moving into and out of your lungs, only about 20% of that is actually reaching the alveoli and participating in gas exchange. So, here's an example of that, how the influence of breathing pattern influences the relative matching between alveolar and minute ventilation. So, again, alveolar ventilation depends on breathing pattern, and I think it's clear that minute ventilation does not always reflect alveolar ventilation, because alveolar ventilation is a function of both minute ventilation as well as dead space ventilation, which is determined by breathing frequency as well as the sum of anatomic and physiologic dead space. So here are three arbitrary breathing pattern examples. We have a rapid and shallow breathing pattern, what we'll call a normal breathing pattern, and a deep and slow breathing pattern. The key here is, look, the minute ventilation under each circumstance is identical, 6 liters a minute. You breathe 150 mLs at 40 breaths a minute in rapid and shallow, 50 mLs or half a liter times 12 breaths a minute in the normal, and a 1 liter breath by 6 breaths a minute. In each case, the minute ventilation is 6 liters. But because the alveolar ventilation is the minute ventilation minus the dead space ventilation, you can appreciate here that if we have the same anatomic and sum of anatomic and physiologic dead space, okay, let's say for arbitrarily it's 150 mLs, is that the influence of breathing rapid and shallow will result in, at this 6 liters a minute ventilation, will result in a dead space ventilation of 6 liters, and therefore the alveolar ventilation will be negligible. This is essentially like panting. You're just moving air into and out of the diffusible portion of the lung, and no breath is large enough to actually overcome the sum of the physiologic and anatomic dead space and push fresh gas into the alveoli. You're basically just moving it, the nose, the trachea, and the main upper airways. Okay, so kind of like, it's like a dog chasing its tail. It does work, but it goes nowhere. The normal breathing pattern you can see here has relatively better matching between minute ventilation and alveolar ventilation, but if you breathe deep and slow, you can see a near perfect matching between alveolar ventilation and minute ventilation. So again, the bigger the breath, the more delivery of fresh gas to the lung. So, with everything that we know, what would be the optimal breathing pattern during exercise to try to facilitate gas exchange? You would like to breathe as deep and as slow as possible, right? Because if you breathe fast, you amplify the effect of dead space on alveolar ventilation. So you would like to breathe really big breaths, doing it relatively slowly. Unfortunately, that is the adaptation that most of us will adopt. We will tend to increase tidal volume early in exercise, and once we reach that balance, somebody asked the question last class about, do we just basically take vital capacity breaths, during heavy exercise? No, because again, there's that offsetting effect, is that if you took every breath all the way down to residual volume and every breath to total lung capacity, you're forcing your lung to breathe against the high resistance at low lung volumes and against the high elastance at high lung volumes. So what you do is that you might compromise a little bit of alveolar ventilation by breathing optimally in terms of depth, which turns out to be about 50 to 60% of tidal volume expansion relative to vital capacity. But then you will facilitate increases in ventilation to try to match metabolic demands, which we now know are very important, by increasing breathing frequency. Okay, so again, this is probably a bit of a cost sharing or adoption where we're trying to now minimize the work of breathing, even if it's at the expense of not completely optimizing alveolar ventilation. Because maybe the body's so in tune to the effect is that the negative consequences of an elevated work of breathing are more detrimental than the potential positive consequences of taking bigger breaths and the impact on alveolar ventilation. I don't know if that's true, but it seems to make sense that that's what the body is intuitively doing. It's choosing the lesser of the two evils. So, you can see the discrepancies at given minute ventilations by changing breathing pattern. So, a brief review of gaseous exchange in the lungs and the tissues, because obviously it is important to know how oxygen and carbon dioxide move and are transported. So, diffusion of gases. Dalton's law dictates that the total pressure of a gas mixture is equal to the sum of the pressures that each gas would exert independently. So basically it's the sum of partial pressures of the individual gases in a mixture. So the partial pressure is calculated as the percentage concentration of a specific gas multiplied by the total pressure of a gas mixture. So, percentages and partial pressures of gases in dry ambient air at sea level. So, for example, the air that we're breathing right now. If we look at oxygen, carbon dioxide, and nitrogen, we know that oxygen is about 21% in the inspired air at a sea level partial pressure of 760 millimeters of mercury, gives us an atmospheric partial pressure of about 160 millimeters of mercury. Carbon dioxide, very low percentage in the ambient air, multiplied by the partial pressure converted to a percentage, multiplied by a partial pressure, you can see very, very low partial pressure in the dry ambient air, and the majority of which that we have contributing to the 760 is coming from nitrogen and other inert gases that aren't listed there. When the air moves from the atmosphere and into the respiratory system, it basically becomes humidified and moistened. And so now we have another factor that's exerting a pressure, which is going to be basically the moisture that we need to take into account, the water vapor pressure, okay, of this humidified air. So you can see now that oxygen, again, is being inspired at the same percentage, But now we have to account for the fact that within that total partial pressure, we have to account for the water vapor pressure, which is now fixed at 47. And so the multiplication factor is now, to get moist tracheal air, is now 713 millimeters of mercury, which is less than atmospheric partial pressure. And so you can see is that as soon as the air becomes basically moistened in the upper airways and humidified, we actually lose some partial pressure of oxygen. So as it's transporting from the atmosphere and into the body, we actually desaturate the amount of oxygen in the inspired air a little bit. And this is also true for nitrogen, because part of that partial pressure is being exerted now by the water vapor pressure at the same percentage concentration, according to Dalton's law. So, the exchange of oxygen and carbon dioxide between the lungs and the blood and their passive movement into and out of the tissues conforms to Fick's law of diffusion. We also know that Henry's law states that if temperature remains constant, the quantity or mass of a gas that dissolves in a fluid will vary in direct proportion to its partial pressure. So again, the higher the partial pressure, the greater the dissolving factor. So two factors ultimately govern the rate of gas diffusion into a fluid, for example, the blood. The pressure differential, so that pressure gradient, the driving pressure, between the gas above and below, between the gas above the fluid and the gas dissolved in the fluid, as well as the solubility. So you can appreciate that, for example, if we have a circumstance where we have a high pressure differential, we'll have a higher rate of diffusion. As well, if the solubility of the gas. So the solubility of the gas in a fluid, where the solubility reflects the quantity or mass of a gas dissolved in a fluid at a given partial pressure. So again, certain gases have different levels of solubility. Gas with high solubility has a high concentration at a given partial pressure. So carbon dioxide is a much more soluble gas than oxygen at a given partial pressure. So as a result of this, carbon dioxide actually moves much more freely into and out of the body fluids down a relatively small pressure gradient. So we know that we have relatively narrow pressure gradients for carbon dioxide between the lungs and the blood. We have to have